Okay, we're going to go ahead and get started here and try and stay on schedule, as one always optimistically hopes at the start of the meeting. So welcome to the 2018 Genome Engineering Workshop hosted by the Joung Lab. My name is Rhiannon. I'm a member of the Joung Lab and one of the organizers for the meeting. And we are delighted to have you guys all here. And our real goal is basically to teach you as much as we can in the next day and a half and answer as many questions as you can. So you may have seen the slide floating past. Most of the people from the Joung Lab are clearly demarcated in our gray shirts. So if you have questions, find someone in a gray t-shirt and we will do our best to get your questions answered. So the format for today, we're going to have 3 talks from people from the Joung Lab, and then we'll have breakout sessions after that where you guys can get one-on-one help and advice on your experiments. And then we'll have the keynote speaker Eugene Koonin this afternoon, and then we'll have poster session with beer and snacks. So you should definitely try and stay all afternoon if you can. I know it's Mother's Day. I'm a mother. I appreciate anyone that came, even if they maybe had family obligations, and we'll definitely try and make it worth your while. So with that, I'd like to introduce our first speaker. Linyi is a graduate student in the lab. He's going to talk about just the basics of CRISPR and kind of introduce you guys to all of this and get things started. And I can see he's wearing his fancy shoes today, so I think you're going to get a really good talk. Thanks, Linyi.
Thank you very much, Rhiannon, and thank you all so much for coming. If you're coming from out of town, welcome to Cambridge, and very happy Mother's Day to all of you. So as Rhiannon mentioned, I'm a graduate student in the lab, and today it's my privilege to talk to you about some of the work we've been doing on CRISPR-Cas systems, and in particular, genome editing with Cas9. A bit of an echo. Can everyone hear me okay in the back? Yeah, okay, great, thank you. So today I'd like to talk first a little bit about just the basics of using CRISPR-Cas9 for genome editing in the lab, and then highlight a little bit of the frontiers in current development of CRISPR technology. So I think the story starts with the sequencing of the human genome almost 20 years ago now. And since then, the cost of sequencing genomes has really exponentially decreased, especially with the advent of next generation sequencing technologies in the past 10 years or so. And with that, we've accumulated a wealth of data about the genomes of all organisms in all kingdoms of life, and in particular about our own genome. And in the process, we've gained a lot of insights into disease processes, such as some of these shown on the slide here. But there's also much to be learned still. Even though we're very good at reading, we don't necessarily understand everything that's encoded in the genome and what that means and its implication for biology. In complement to a lot of these genome sequencing projects have also come a lot of detailed insights from genome-wide association studies to map out correlations between different kinds of variables. And I think what is still kind of an open question and still something to be solved is the question of causality. What genes actually cause what kind of phenotypes, and how do we systematically interrogate that? And I think to do that required the development of a new technology that we can actually use not only to read genomes, but to manipulate them in a targeted way. So the idea of doing genome editing actually is quite an old idea, over maybe about 30 years or so, which is that if you create a double-stranded break somewhere in the genome, the host enzymes that are in the cell repair this double-stranded break in such a way that really facilitates genome editing. So if we start out with a break somewhere in the genome, typically one of 2 things can happen once that cell recognizes the break. So in the first pathway is called non-homologous end joining, which the cell basically uses to recognize this break and go into a sort of panic mode and tries to stitch together the break in any way it can to prevent it from dying. And in the process, it has a propensity to create these insertion and deletion mutations, really small, like 1 to 20 base pairs or so, right at the break site. And if that happens to happen in a protein coding gene, you can knock out the gene and inactivate it. The second pathway, which is typically less prevalent and less favored in cells, is a homology-directed repair. In this case, in addition to the break, the user also supplies a template sequence which matches both sides of the break, but the middle here, shown in blue, is different. And with some efficiency, the cell can actually recognize that and use that as a set of instructions to repair what happened in the break according to what was there in the template. So that's a way to create more precise genome edits than non-homologous end joining. So the question is, how can we create this double-stranded break in the cell? We know that's very useful, but the genome is 3 billion bases long, and it's kind of like finding a needle in a haystack. So in the early days, people were using designer proteins. So these are proteins that actually have protein DNA-specific recognition to be able to target specific sequences in the genome. These were very expensive. As of 2012, maybe $5,000 to create a novel one for some custom designer nuclease. Early generations one included zinc finger nucleases that recognize kind of 3 bases at a time based on protein recognition. And a bit later on, talins, which have a one-to-one kind of base correspondence with these 2 hypervariable residues encoded in a modular protein architecture. So this is extremely useful and enabling, but it also is a little bit restricting because for every new target site, you need to engineer a totally new protein. So here's where CRISPR comes in, which is an RNA-guided DNA recognition module that's actually an anti-phage system in bacteria. It's a bacterial immune system that actually stores a part of the viral DNA that's from a previous infection, and then uses that as a reminder for future infections to actually cleave the viral genome. So this is kind of an actual image of it in a cartoon for how CRISPR works. And at the heart of CRISPR is a protein RNA complex called Cas9 here, shown in this cartoon. And Cas9 is an RNA-guided nuclease that actually targets the phage genome. So in the recent years, there's emerged a pretty comprehensive categorization of different kinds of CRISPR systems out there. And there's roughly kind of one watershed between the different kinds of CRISPRs. One in which kind of the effector function to actually target the phage genome is split over multiple proteins. This is class 1. And class 2, which constitutes Cas9 and some other proteins, has a single large effector protein that's a lot more amenable for adaptation and harnessing for genome editing because we don't have to worry about all these proteins coming together, just a single one. So I'm going to talk for the rest of the talk on class 2 systems. So as I mentioned, the most well-known of these proteins is Cas9. So this is an RNA-guided programmable protein that cuts DNA. This is the target DNA strand, and Cas9 is this protein in yellow, and it binds to a piece of guide RNA that consists of a variable region that is actually complementary to the target site that we want to edit, and a constant region that's required for activation of the Cas9 nuclease. So the guide RNA invades and unwinds the target and tries to look for sites where it's perfectly complementary. And when it finds a site like that, and it also has the secondary requirement that this sequence right next to it called the protospacer adjacent motif, or the PAM, in this case 2 guanines, is also there. So if we have complementarity and we have the PAM, then Cas9 unwinds the DNA with the guide and makes a cut in the DNA. So we can create targeted double-stranded breaks this way. So of course, the main advantage of Cas9 is we can keep the protein the same for all experiments and only change the guide RNA, which is much easier than engineering a totally new protein to do engineering. So that's the protospacer adjacent motif. So here is just a quick illustration of how that works in the cell. So here Cas9 has been put into the nucleus, and it finds the PAM sequence. And it uses a guide RNA, here shown in red, to unwind the target DNA. And if there is perfect base pairing, as in this case, where all the bases match, then Cas9 actually creates a double-stranded break in the DNA. That break is then recognized by host factors, and there's some shuttling and lots of things going on here. But at the end, there might be some mutations at this target site right near the break shown in red, which then can activate the gene of interest. So the cleavage activity of Cas9 is not the only function. Turns out Cas9 has 2 nuclease domains and 2 key active site residues in each of these domains. And if we actually inactivate them with alanine substitution mutations, this can be turned into something that only binds but doesn't cut DNA, which is an alternative and useful platform for recruiting stuff to DNA but not actually cutting it. So that's kind of the basis for CRISPR-based genome engineering, using Cas9 as a nuclease of some kind for genome editing, and as well as Cas9 as just a DNA binding programmable module without actually cutting. And some other very interesting developments more recently that don't fall in these categories, which I'll touch a bit on later. So in the past couple of years, this has enabled a lot of applications in many domains of biological science, from engineering organisms and crops to being able to allow us to systematically interrogate the consequences of genetic variations in research. So I want to spend the rest of the talk first on kind of outlining a prototypical CRISPR experiment and some of the design considerations we might put into that, and some selected frontiers in more recent years in how the CRISPR toolbox has been expanding. So this is a prototypical workflow for generating a model cell line, either a knockout or a knock-in, using CRISPR-Cas9. So we first start with kind of designing and constructing and thinking about the reagents and the guides that we want to deliver. Constructing the reagents and thinking about the delivery method, whether it's a plasmid or a virus or the protein itself. And then actually doing the CRISPR experiment and functionally validating and obtaining a cell line of interest. So I want to first talk about the design of this experiment and some of the things we might think about for designing a CRISPR knockout. So arguably, here are 5 of the main strategies we can use to modify genomic sequences with CRISPR. These differ by the number of guides that we deliver, as well as the pathway in the cell that's responsible for affecting the change. In addition, whether or not we supply a donor sequence. So in the first case, we actually don't create any double-stranded breaks. This is a new technology developed by David Liu's lab and others around the world, where we actually directly change a single base from either a C to a T or an A to a G. So delivering one guide called base editing, and it doesn't use a double-stranded break. 3 different strategies with a single double-stranded break and a single guide, either knocking out a gene by just creating a cut and letting NHEJ take over to get a mixture of insertion and deletion mutations. We're doing the same thing except supplying a donor to try to specify the kind of mutation profile that occurs. And then using NHEJ with a large donor that actually gets ligated into the break site to insert a large cargo into a specified break site. And then finally using 2 guides to delete a segment of the genome in a programmed way. So there's some considerations for actually designing the guide sequences or guide sequence for each of these strategies. Some general considerations are the cleavage efficiency and the off-target activity, which is applicable for all of these strategies. And I also want to talk about some special considerations for choosing guides for each one of these strategies. So there's a lot of great work done here and elsewhere to understand what makes a good guide, what makes it efficient, and what makes it specific. And a lot of that has been codified in a lot of online software. And there's many software available that's highly valuable. I want to highlight one of them that I'm particularly fond of, which is a tool in Benchling. It's an integrated on-target and off-target prediction tool, which kind of integrates the state-of-the-art on-target and off-target scoring in an integrated workflow where you can have it automated, in an automated fashion, pick guides for you and tell you kind of what the scores are and where it is in the target site of interest. I think one thing that's important to note is even though these are very carefully thought out, these scores are really approximate, and they're not a definitive kind of measure of how the guides actually work. So you can oftentimes find guides with lower on-target scores that are actually more efficient when you test them out in the lab than things with high on-target scores, and vice versa. So it pays to kind of look broadly and just create a list of things that you might try out. As I mentioned, there's also some more specific considerations for each of these strategies in terms of what makes a good guide. So I want to talk about just knocking out a single gene with a single guide by indels. So it turns out that not only the efficiency of the guide is important, but also what kinds of indels you get after you deliver the guide is also really important. So if you're trying to knock out a gene, you're trying to create lots of frame shift mutations instead of in-frame mutations. So picking a guide that will give you frame shift mutations, for example, might be something that's really important. So a lot of work in recent years has highlighted that even though you create a double-stranded break in the genome, the kinds of repair that you get is really non-random, and it really depends on the sequences that are there right next to the break site. So here is a particular Cas9 target with the ends indicating the variable regions and the red triangle indicating where the break is happening. And I'm going to number these bases just for future reference, starting from the 3' end to the 5' end, so base 1 to base 20. And the highlighted base here is the 4th base, shown in red. So one of the observations that we and others have made is that the kinds of mutations you get actually depend on what base is there at base 4 in the target site. So here's some data showing that. So this is a bit of a busy plot, but to take you through this, on the x-axis is the percent indel, so the efficiency overall of getting cuts in the genome. On the y-axis is how many insertions, or among the indels, how many of them are actually single base pair insertions. So this would all be kind of frame shift levels. And each dot is a different guide sequence in the human genome. And this is stratified by the identity of the base at position 4. So if we have a G or C at position 4, most of the guides don't have a very strong level of these single base pair insertions, but kind of an interesting observation is that if you have an A or T at the 4th base, then the percentage of the probability of getting a single base pair insertion is much higher. So it just might be like a slight design consideration that if you're trying to get a lot of frame shift mutations, if you pick an A or T at this base, it can help. The second consideration is microhomologies that are present at the target site. If you have bases that are very similar on each side of the target site, there's a very strong tendency to collapse those microhomologies into a single one. So here, for example, the break is shown in green, and there's 2 microhomologies, TCA on both sides. With probability much higher than random, a lot of the repair outcomes will actually just be deleting everything in between the TCAs and getting a single TCA. So if these TCAs are positioned in such a way that it gives you an out-of-frame mutation once you collapse the microhomology, then that can be used to your advantage. And there's a great prediction tool at this website that you can actually use to predict if there are strong microhomologies or not near the cut site. The second consideration for a different strategy, so instead of using a single guide to get a mixture of indels, to use that one guide and also a donor template to create a what-defined cell line, there's also a consideration where the distance of the guide on the cut site to your intended mutation of interest actually plays a big role in how efficient things turn out to be. So here is a paper in 2016, a great work, demonstrating the effect of distance to the cut site to HR efficiency. And there are 3 categories here, either to get double mutants, so homozygous lines, a wild type line, or a heterozygous line. And there's kind of a different optimal distance for each case. So if you're trying to get a homozygous kind of knockout line or knock-in line, then it's best to choose the cut site to be as close as possible to the intended site of mutation. But if you're trying to get a heterozygous one, it's actually maybe a little better to move it farther away, because if your guide is too efficient, then maybe there are too many homozygous clones that you obtain. And there's kind of a trade-off there. So maybe moving it out about like 10, 12 bases or so can help for obtaining a heterozygous line. So to summarize all of that, I think for general considerations, online tools like Benchling are really good at providing general guidelines for what's a good guide in terms of on-target and off-target activity. And there are also strategy-specific considerations. For knockouts, checking for microhomologies or an A or T at the 4th base, and for HDR, picking guides with a good distance to the site you want to mutate. Okay, so this is kind of the roadmap for the first part of the talk, and I'm going to move on to the second point, which is constructing reagents and thinking about how to deliver CRISPR into the cell line or animal model of interest. So these are arguably the three most popular ways to deliver Cas9 into cells. So either using plasmid-based vectors or lentivirus or AAV. So lentivirus will give you kind of stable integration into the genome at a more or less random location. AAV is really good for in vivo delivery for a live animal. And then protein delivery also is very good for having very low off-target effects and low toxicity. For plasmid delivery, there are many, many reagents available on Adgene, which is a great resource. This is a highlight of some of the more popular reagents from our lab. The PX330 series is just expressing Cas9 and guide on a single backbone. 458 and 459 are extensions of PX330, where in addition to Cas9 and guide, it either has a GFP or a puromycin resistance selection marker. There's also a lentiviral vector to package lentivirus with Cas9 and guide. And then an additional vector that has a different species of Cas9, which is a bit smaller and better packaged into AAV. And Adgene has great resource pages to to read more about on these. So after delivering the reagents and obtaining knockouts or knock-ins, we want to assess how well the editing actually happened. So this is the functional validation and further characterization. So to my knowledge, there are three main ways to assess how much indel you're getting from a CRISPR experiment. The three ways are using gel electrophoresis, Surveyor, or the T7 endonuclease 1 assay. The second method is using Sanger sequencing, method called TIDE, which I'll talk about in a bit. And the third, and our favorite one, since it's probably the most sensitive, using deep sequencing to actually sequence the kinds of mutations that happen at the locus of interest. So I'm going to just briefly go through each one of these right now. So Surveyor is a method to quantify indels using gel electrophoresis. So we start off with a mixture of genomes, some of which contain mutations and some of which don't. And we want to figure out what percentage of the genomes actually contain mutations. So here's an indel here and some amplicons with no indel. So if we amplify this entire mixture, we get a mixture of amplicons. And then if we melt them and actually re-anneal them to each other, instead of the red one and the red one re-annealing, we actually get some percentage of the red one and the black one re-annealing, and so forth. When we incubate this with the Surveyor nuclease, or the T7 endonuclease 1, it actually recognizes bulges like this, where there's not a perfect base pairing, and it makes a cut wherever it forms this sort of bulge. So this is a way to distinguish between stuff that re-annealed correctly and stuff that re-annealed with some bulge in it. So once the nuclease cuts, we actually get two fragments. If there was a bulge there, then there's two fragments exactly where the CRISPR editing site happened. If we run that on the gel, we can see the fragments at defined locations relative to the uncut band. And by quantifying the intensity of the fragments, this is a good way to assess editing efficiency. The second method is TIDE, or tracking of indels by decomposition. This is a similar idea as Surveyor, except we're not using the Surveyor nuclease. Instead, we're using Sanger sequencing to kind of deconvolute what's edited and what's not. So if we start out with a mixture of products, some of which have edits and some of which don't, and we just Sanger sequence the entire mixture, if there are no edits, then it looks clean all the way through. And if there are edits, it looks clean up to a certain point, and then we hit the editing region where there's insertions and deletions, and stuff just gets super messy. What's really cool is that by using a sophisticated computer algorithm, we can deconvolute the types of sequences that actually are responsible for making up this mixture and kind of get an estimate of the different kinds of sequences that are actually in the mixed population. The third strategy is using deep sequencing. So here we take the entire region, PCR the mixture, and then actually just sequence a single molecule and then count how many amplicons in the sequencing reads have indels and how many don't. So what do we use? What's the best thing to use? I think there are advantages and disadvantages to each one. So these are definitely much faster and probably more amenable if you have a small number of samples. So Surveyor and TIDE typically give maybe a 1% to 2% level of sensitivity. Surveyor tends to underestimate the types of indels because the types of mutations there are not totally random. So you don't always get cleavage even if there's a mutation there. I think probably if you have access to deep sequencing, we found that it's the most sensitive strategy. The downside is that it requires pretty high throughput, so it might be hard to get kind of a single sample into a MySeq run to quantify that way. For general, just assessing overall how well things worked, I think Surveyor and TIDE worked pretty well for a small number of samples. Okay. So now I want to talk a little bit about some frontiers in recent years in expanding the genome editing toolbox, the things other than CRISPR-Cas9. So the first application is actually a little bit old, back in 2014, where our lab created a Cas9 mouse. So this is a mouse that actually just has Cas9 in there already. And then if you just deliver the guide RNA to that mouse, it can start creating edits by itself. So here is shown kind of a Cas9 mouse where there's one guide delivered, and you can see accumulation of edits at this particular locus, and also kind of phenotypic validation. So this is a way to accelerate disease modeling inside a live animal. CRISPR also enables high throughput screening, so not just targeting one guide at a time, but doing it on a genome-wide basis. Here's one example of that. So this is a patient with melanoma that's been treated. So here you can see some tumors all over the skin. And after 15 weeks of drug treatment, you get a profound phenotypic reversal where most of these mutations or most of these tumors kind of go away. But unfortunately, after a few more weeks of treatment, there's this relapse where tumors start coming back again. And the molecular mechanism underlying this process, some of which is still unclear. So one thing that CRISPR can do to enable understanding of a system like this is to systematically interrogate every gene in the genome to ask what might be responsible for causing this kind of resistance. So here we can actually use oligoarray synthesis, which is fairly inexpensive, to create a library of guide RNAs that target every gene in the genome, and clone that into a pooled library of lentiviruses, which are then used to infect these cells and kind of screen in a pooled way what's giving rise to resistance and what's not. So here the guide RNA acts both as a barcode for what gene got edited, as well as the vehicle for actually editing the gene. So here's that same experiment done in a cell line model where we're screening for what kind of genes are giving resistance to that drug. And we find that there's this collection of genes up here out of all these genes genome-wide that are screened that are highly enriched and possibly confer some pathway modifications that confer resistance. So Julia's talk will be next, and she'll talk about kind of all sorts of different screening processes and both activation and knockout and all the different variants that are possible today. One set of recent developments is also not only just using wild-type Cas9 that came directly from bacteria, but also engineered Cas9 variants that have different properties that allow us to target DNA differently. So one theme of research has been engineering the Cas9 protein itself so that it's inherently more specific at targeting DNA. To the best of my knowledge, there are currently 7 different high-specificity Cas9 variants out there that are very great work and very useful. So starting with kind of the first 2 that were published in 2016, ESP-Cas9 and SP-Cas9-HF1. And then more recently, additional variants that also have fantastic specificity. As before, all of these variants are available on Adgene. And I think, yeah, so these are really good ways to, if you're concerned about off-target effects, to reduce off-target effects. It's also been our experience that for at least some targets, at least some of the variants have a tendency to lose a little bit of activity. So one consideration for using a high specificity Cas9 is that you might want to test a couple more target sites to make sure you're getting a guide that actually works really well. The second consideration is that usually these Cas9s, with the exception of Stiper-Cas9, only work well with a fully matched 20 base pair guide sequence. So if you're trying to introduce kind of a mismatch at the 5' end to use with the U6 promoter, that doesn't really work very well with a lot of these, and especially these first 3 up here. So using a fully matched 20 base pair guide is essential. Cas enzymes have also been engineered to be a little bit better at recognizing different DNA sequences in terms of broadening the PAM recognition motif. So here are 2 variants of SP-Cas9, 1 variant of SA-Cas9, and 2 variants of another enzyme, CPF1, which I'll talk about in a bit, that actually can recognize PAM sequences that were not originally able to be recognized in the bacteria. So it's great work from Dr. Keith Jung's group at MGH, which you might hear a little bit about tomorrow. Engineering variants of SP-Cas9 to recognize not only NGG, but NGA and NGCG PAMs. And SA-Cas9 to recognize NNN or RRT PAMs. So these significantly expand the number of target sites that are able to be hit in the genome. More recently, we've also done some work on engineering the specificity of another enzyme, CPF1, which I'll talk about, to recognize this TTCV and TCCV PAMs and TATV PAMs, in addition to the triple T that it recognizes normally. So as I mentioned earlier, one really exciting new development is the ability to edit single bases in the genome without actually creating a double-stranded break. So this is work highlighting, slide highlighting some work from Dr. David Liu's lab here at the Broad, using an engineered Cas9 nickase fusion to actually adenosine or cytidine deaminase domain. And this actually unwinds the DNA. And instead of making a cut, it uses the deaminase to actually pull off an amine group from either A or C. And that converts that base into a G or a T. So to make point mutations, which is sometimes a large effort, this can significantly accelerate the time and the efficiency of doing an experiment like that. Okay. So finally, I want to highlight some work on expanding the CRISPR toolbox to not just Cas9, but other kinds of CRISPR systems that are out there. So in the last couple of years, we've really come to the understanding that CRISPR systems are really diverse. It's not just one Cas9, but many Cas9s, and it's not just Cas9, but many other kinds of enzymes that are more diverse than that. So there's been a consensus that there's now 3 kinds of class 2 CRISPR systems, 2 that target DNA and 1 that targets RNA. So the 2 DNA targeting systems, one of which includes a large family of Cas9 enzymes, and then a second one that includes Cas12, one of which is CPF1, which has been harnessed for genome editing. In addition to the 2 DNA targeting systems, there's a third system that actually targets and cleaves RNA instead of DNA. And Max will talk a little bit about that later this afternoon. So I want to talk a little bit about some work on CPF1. So CPF1 or Cas12a is an alternative editing enzyme relative to Cas9. So here is the genetic locus in the bacteria for Cas9 versus for CPF1. And you can see some major differences already. There's a difference in the architecture of the protein itself, as well as in the architecture of the neighboring genes and the RNA around. And this can be used to advantage for genome editing. Here is a comparison between Cas9 and CPF1. So Cas9 recognizes an NGG PAM right after the target site. And it contains 2 nuclease domains and a relatively long piece of RNA, the guide RNA. In contrast to that, CPF1 actually has a PAM on the other side of the target site, on the 5' end. And instead of being a G-rich PAM, it has a T-rich PAM, so 3 Ts instead of 2 Gs. And what's interesting is that because it doesn't require the tracer RNA, the guide RNA is much shorter. And because of that, it's a lot easier to deliver guides with CPF1. In addition, Cas9 creates a blunt end cut, whereas CPF1 creates a staggered cut with a 3 to 5 base pair overhang. So one of the advantages of CPF1 is actually being able to take advantage of the fact that it has a much shorter piece of RNA. So here's Cas9. This is the pre-CRISPR RNA that's transcribed, and it's hybridized with a bunch of copies of the tracer RNA. And that's required for getting this pre-CRISPR RNA processed into individual mature CRISPR RNAs that can actually target a particular target site in the DNA. This requires Cas9, and it also requires the host factor RNase 3. In contrast with CPF1, this is a lot more simplified because we don't have this tracer RNA requirement. And it turns out that if you just put the CRISPR RNA array with CPF1, CPF1 itself can just cleave this into individual guides. So this is cool, but why is that useful? So this is useful because it allows us to target multiple genes at the same time in an efficient manner without delivering guides all at the same time. So here's an experiment that Bernd and Matthias and others in the lab have done to demonstrate the utility of this feature. So here is an editing experiment where we have the expression of CPF1 in human cells, as well as an array of guides right before the CPF1. And this is an array of 4 guides that targets 4 different genes in the human genome. And it's delivered in one of 4 different formats, either as single guides with each guide driven by its own promoter, or as just a big array driven by a single promoter at the very beginning. And there's 3 configurations of the array, depending on the lengths of these direct repeats and the guides. And the takeaway that's very interesting is that with this shortest configuration, array 1, the targeting efficiency of each of the 4 genes is basically the same as if we deliver each guide separately with its own U6 promoter. So this is a much easier configuration to program and to clone and to make sure no recombination happens. So just by delivering this one array, we can simultaneously target 4 different genes. In addition to CPF1, the alternative DNA targeting system, there's also a third class 2 CRISPR system, the RNA targeting system, Cas13a and Cas13b. So one of the applications that we can build off of targeting RNA instead of DNA is actually to edit the RNA instead of to edit the DNA. So this is kind of a version of RNA-based editing that our lab developed recently, where we can take A in the RNA, adenosine, which has this amine group, and if we can convert this A into, if we can basically remove this amine group and replace it with a hydroxy, this A gets converted into inosine, which is recognized as guanosine in the cell. So if we can convert A to I, that's effectively an A to G edit in the RNA. So how do we do that? We can use a Cas13 here as a module to target the RNA, and then fuse on an adenosine deaminase domain to the Cas13 that will then recognize the base that's kind of programmed to recognize the A, and then deaminate that into an I, which is then recognized as a G. So how efficient does that happen? So these are different genes in which this editing has been attempted, and you can see very efficient editing of targeting guides up to about 30%. And Max will talk a lot about this later on in the afternoon. So I want to end on one thought, which is that there's been a sort of continued search for technologies, and we've learned a lot from the natural diversity of CRISPR systems that are already out there. We've learned that there's 2 different kinds of very diverse families of DNA targeting, DNA cleaving enzymes in CRISPR, as well as 1 RNA targeting system. But in the last 10 or 15 years, we've also got the ability to sequence a lot more microbial genomes than what was available kind of when CRISPR was beginning to be discovered. So even in the last 4 or 5 years, we've seen, in terms of the number of sequenced microbes, kind of this exponential increase, and most of it has been in the last couple of years. So I think a really exciting question that we're wondering about is, if we can discover things as powerful as CRISPR in microbial genomes, could there be other systems out there that might confer maybe different kinds of functionality that might also be very powerful biotechnology? And so there might be opportunities for kind of sequence mining and discovering of new protein effector systems in these genomes. So I'd like to thank you all very much, and I would like to thank everyone in the Joung lab, and I'd be happy to take any questions. Yeah.
So for the CPF1 cutting, is the quantification of its nuclease activity done similar to Cas9 using like Tide or Surveyor EC?
Yeah, that's a great question. Yes, so CPF1 edits also create indels at the break site that can be quantified with either of those methods, or any of those 3 methods. One important feature to keep in mind with CPF1 is that it almost exclusively creates deletions instead of insertions. So Cas9, you oftentimes get a 1 base insertion. CPF1, you almost never get that. So maybe 98% of the repair outcomes will be deletions. That doesn't affect Tide or NGS very much. It actually has a side effect of making Surveyor a little bit more accurate than for Cas9. So Cas9, it has a tendency to maybe underestimate a little bit. With CPF1, it tends to be a better reporter for efficiency. So, yeah.
So is there any known like a ratio in between the guide RNA and Cas9 that is working better? And the other one is like, is there any known stability of Cas9 and then our guide RNAs overall?
The optimal stoichiometry between the guide and the Cas9? Yeah, so I think at least the protocols that we are accustomed to following typically maybe give a little bit of excess of guide relative to Cas9 just to make sure the complex is kind of fully formed. But I think as long as there's a good stoichiometry where you're pretty sure that you're getting, excuse me, stoichiometry where you're pretty sure you're getting a lot of the active Cas9 guide complex forming. I think it's a, I think the experiment should be pretty robust to that. Yeah.
Another question. So except maybe like in addition to the UC promoter, any other like a more robust promoter has been tested?
Yeah, we found that the U6 promoter works pretty well. There's alternative promoters that actually have a tRNA promoter. So instead of a U6 promoter, you can put a tRNA in front of the guide, and that kind of gets processed in a way that maybe is more versatile because it doesn't have the requirement of the G. So with the U6 promoter, you want your guide to start with a G, but with a tRNA promoter, you can get away with guides that don't start with that and not have to change it to a G. But I think for a lot of purposes, the U6 is kind of the gold standard, and it works really well.
Yeah, I'm curious whether any study has been done. So for a specific cut site in the genome, if there are 2 copies of the cut site in genome, how many molecules of the CRISPR-Cas we would need to give to the cells so that they efficiently scan throughout the genome and identify the cut site?
So how many molecules of Cas9 do you need in any individual cell to create? You know, I don't have an accurate answer for that. I think maybe the best answer I have is for protocols that deliver ribonuclear protein complex directly to the cells, and typically just kind of measuring the concentration of overall protein that's delivered to the well, and if you divide that by the number of cells and kind of estimate the delivery efficiency, that might be a good estimate for how many molecules are actually getting in. So yeah, I guess I don't have a hard number I can have at the top of my head right now. But.
Great talk. When you were explaining the position effect in the guide RNA, you show guanine as the 20th nucleotide. Is there any specific reason that you put that in?
Yeah, I think that's really only applicable for when you're delivering an expression cassette that drives expression of the guide rather than the guide directly. So it turns out the U6 promoter basically requires a G at the very beginning to begin transcription. And if you don't have that G there, it either doesn't transcribe well at all, or it maybe just starts transcription at a site that's a little bit different, and so your guide might get artificially truncated. So it's just because of the U6 promoter.
What's the effect of chromatin packing, the density of chromatin within the nucleus on efficiency of the targeting for euchromatin versus heterochromatin?
Yeah, that's a great question. I recall there was a fantastic paper by Dr. Jonathan Weissman's group where they found that nucleosomes actually impede CRISPR-Cas9 activity significantly. So yeah, I think if the chromatin is highly compact and there's just kind of lots of stuff there, then Cas9 is not as good as finding and unwinding it. And I think that's consistent with what we've seen for regions of open chromatin, kind of the editing efficiency definitely is higher. So I guess if you have a choice of target sites, and one is kind of right where some nucleosome is sitting, and another one is maybe farther away, then it's probably good to take that into account too. Yeah.
Okay, we're gonna move on. We're going to move on now in the interest of time, but Linyi will be here during one of the breakout sessions and available to answer lots more questions. So our next speaker is Julia. She's a graduate student in the lab. She's a fourth-year graduate student who is our master of screens, and she will do about a half-hour overview on using CRISPR for large-scale screens.
Thank you for the introduction. Thanks everyone again for coming out here for our annual Genome Engineering Workshop. Today I'd like to give a brief overview of how to set up a genome-scale CRISPR screen. Most of this is in a protocol that we published last year, so I won't go into the details. Instead, I'll go give a overarching, talk on like how to just the main points that you need to think about when setting up a CRISPR screen. So to begin, the forward genetic screens are very powerful tools for mapping genotypes to phenotypes. Traditionally, this was done by chemically mutagenizing flies, picking out the flies with interesting phenotypes, such as white eyes or mutations in the antennas, and going back and trying to figure out which genes, which essential developmental genes actually contributed to the phenotype of interest. Now, recently, with the development of shRNAs, this process has become much quicker and much more easy to do. The way shRNAs work is that the shRNA is processed into siRNA and can knock down base pair complementary mRNAs. Now, in the screening format, by sequencing the shRNA, we can map the targets of the shRNA and figure out which shRNAs contributed to the phenotype of interest. And so in a way, thanks to large-scale next-generation sequencing, shRNA screens have been very useful in discovering a lot of biological processes. The issue with shRNAs mainly is that using shRNAs can be very, very unpredictable. A lot of the times, shRNAs will have a lot of off-target sites, and this kind of thing tends to confound the screening results. So more recently, the CRISPR-Cas9 knockout, as Linyi talked about earlier, has been shown to very robustly knock out target mRNAs by basically introducing a premature stop codon, leading to the depletion of the target mRNA via nonsense-mediated degradation and decreasing the amount of functional protein in the cell. What one can do, because the Cas9 knockout is much more reliable and has lower off-targets than shRNAs, the screening results from CRISPR-Cas9 screens tend to be much cleaner. CRISPR has also been shown to be used as a robust DNA binder by inactivating the two residues that are involved in DNA cleavage. Cas9, we can then add a bunch of DNA activation or repression domains to make Cas9 a robust activator or repressor, respectively. In this case, I'm showing the Cas9 activator developed by our lab that has modified guide RNAs with MS2 hairpins. These MS2 hairpins then bind two additional activation domains, P65 and HSF1. And together, there's 3 activation domains on this Cas9 activator that makes it much more robust than the previous version involving only Cas9 and VP64. So if you target the Cas9 to the promoter sites of target genes, then this can lead to a very robust transcriptional activation, leading to an increase in mRNA production. And as a result, the protein of interest, the levels of the protein of interest is also increased. So together, both Cas9 knockout, Cas9 activation, and Cas9 repression has been used for a lot of different genetic screens. Some of these examples include studying drug and toxin resistance, gene essentiality, more recently, cancer immunotherapy, and non-coding elements. In essence, the CRISPR-Cas9 screens can be used to study any biological process with a screenable phenotype. And I'll go into detail on what exactly we define as a screenable phenotype. So there are 3 main types of screening selections. There's the positive selection, negative selection, and the marker gene selection. If we think about the blue guide RNA as a guide that makes a change that gives us a screening phenotype of interest, in a positive selection screen, the cells with the blue guide RNA will proliferate, and the blue guide RNA will amplify because this is genomically integrated, the blue guide RNA will amplify with the proliferation of the cell. And at the end of the screen, when we sequence the guide RNAs in the pool, we will see more of the blue guide RNAs. In the negative selection screening phenotype, the blue guide RNAs are depleted, and at the end of the screen, we see fewer blue guide RNAs than we started out with. And then finally, the marker gene selection. In the marker gene selection, you either have a reporter line where the screening phenotype is marked by fluorescence, or you can stain with an antibody and followed by also FACS sorting to identify the cells with the phenotype that you want. And in this case, the blue guide RNA will generate a cell that is green. The marker gene selection screens are much more versatile than the positive and negative selection screens, but at the same time, they're also a little bit more difficult to set up because it requires a lot of FACS sorting. So overall, the workflow of the CRISPR-Cas9 screens starts out with making this library for whatever purpose that you want. I'll go into the detail of this in a little bit. Then packaging the library into lentivirus, transducing this lentivirus into cells, and then harvesting and analyzing the screen using various different tools, and then followed by another 4 to 5 weeks of validation. So in total, the screen takes about 3 to 6 months to complete. But at the end, you get an unbiased, systematic assessment of which genes are involved in the mechanism for your target phenotype, making it a very powerful tool. So starting out with library construction. We've provided several different ready-made libraries on Agene. These include the Gecko library, which has the one vector system with the Cas9 and the guide RNA on the same vector, or the two vector system where the Cas9 and the guide RNA are on 2 different vectors. The Naga libraries have 6 different guide RNAs targeting the 5' constitutive exons. In the activation libraries, we have also the 2-vector or the 3-vector system, and they have different components of the SAM activator in each of the vectors. And these tend to have 3 different guide RNAs targeting the 200 base pair region upstream of the annotated transcriptional start site. Both of these libraries come in either the human genome-scale version or the mouse genome-scale version. And I'll point out that even though we have the Gecko v2 libraries on Agene, I would actually recommend going for the Brudello library because that has a much more updated efficiency and specificity score for screening. So we, at the same time, because a lot of screens, especially like non-coding screens or targeted screens, require different libraries than just like the blanket genome-scale library, we also provide different methods for generating custom libraries. So starting out with the custom targeted library, the targeted library is very useful because for instance, in some screens, you want to reduce the number of cells that are required for screening. I will go into this in a little bit more detail, the number of cells that are required for a typical screen, but that number is generally around hundreds of millions of cells. And so for sensitive primary cells, for example, you really have a motivation to reduce the number of cells that are required for your screen. The targeted screens are also very useful for when you are only concerned with a subset of genes. For example, only kinases for a cancer screen. So the way that this works is that we provide a script to isolate a subset of sgRNAs from the larger pool of genome-scale library based on a set of target genes that you want in your library. Then we also have the de novo library design. The Python script for this works is that you have a genomic region of interest, for example, a non-coding region or different, for instance, different long non-coding RNA promoters, and then you can pick guides based on minimizing off-target activity, maximizing on-target activity, and other criteria that helps with sgRNA synthesis. And for this include multiple guides per target and also non-targeting guides. The non-targeting guides help with determining the noise level of your screen and basically determining whether or not the enrichment you see in your screen is a result of noise rather than you're applying a screening selection. And for both the targeted and de novo designed libraries, we provide a very easy to follow protocol that involves basically PCR amplifying the oligos, Gibson assembly into the positive vector of interest, and then purifying this, amplifying this through electroporation. Throughout the custom cloning process and throughout the entire screen, it's very important to maintain representation by scaling up all the reactions accordingly. And we actually have a table in the protocol stating basically for a certain size of your library, how many reactions of PCR you need to do, how many reactions of Gibson you need to do, and how many bacterial clones you need to generate to maintain representation. And then finally, before starting the screen, because the screen is a lot of work, we always recommend that you verify the library representation by NGS, even if it's a library that's already available from Agene. That's because sometimes there's always different biases in amplification, and you just want to make sure that your library is not super skewed, all the guides are there before doing all the downstream work. And our criteria for whether or not you should proceed for the library is basically if you have over 70% of the guide RNAs are perfectly matching to the library, and less than 0.5% undetected guides, and the skew ratio of less than 10. The skew ratio is a measurement of the number of NGS counts for the top 10% of the guides to the bottom 10% of the guides. Okay, the next step for sgRNA screening is packaging the library into lentivirus. For us, lentivirus is usually the most ideal packaging method for screening because it integrates into the genome, which means that for positive and negative selection screens, your guide RNA will amplify with the genome and amplify or decrease with the number of cells in your population. And so you can just measure the NGS count of your guide as a proxy for the number of cells in your population. So in the paper, we include a protocol that has both lipofectamine and PEI. Lipofectamine was what we used for all of our published screens, and PEI we've also found to give comparable results and is much, much cheaper than lipofectamine. So we also provide that protocol. In certain cases, for non-dividing cells, such as neurons, you can also use AAV, but this is much less common for screening. And then after lentivirus production, we will put the library into the target cell line and apply the screening selection. So calculating the lentiviral titer in a screen is super important because you want to keep the lentivirus MOI, or multiplicity of infection, low enough such that most cells only have one guide RNA that is integrated into the genome. This means that when you read out your screen, you don't get a lot of cells that have multiple guide RNAs and therefore multiple different perturbations that can confound the screening analysis. What the multiplicity of infection is it measures the number of cells surviving the selection divided by the number of cells without the screening, the antibiotic selection. And for measuring this, we provide a spinfection method for pretty hardy cells, or the mixing method for things like stem cells and neurons. And this really varies depending on your cell type. What I would recommend is try to spinfect your cells. If they don't die, then do the mixing method. For the sgRNA library, we recommend an MI of less than 0.3. And then after calculating the lentiviral titer, we scale up accordingly. So for scaling up the transduction for the screen, for example, for a library of 100,000 different sgRNAs, if we want a screening coverage of 500 cells per sgRNA, which is what the minimum that we would recommend, and we are transducing at an MOI of 0.3, that means that for each biorep, you need to start out with 167 million cells. So right now, based on this calculation, probably the targeted screens are looking more attractive. So this is a lot of cells, and so you need to plan accordingly because usually for most screens, we recommend at least 2 to 4 bioreps. So that really adds up to hundreds and hundreds of millions of cells, at least to start. And then during the screen, to maintain that representation, after the cells have been already selected for, we just maintain 50 million cells. Okay, so for knockout screening, typically for most screens, people will transduce the Cas9 at an MOI that is fairly high, but less than 0.7. and then introduce the guide library at an MI of less than 0.3. We recommend for this to start the screen 7 days after library transduction, because that's when the indel saturates. So you don't want to start applying your screening selection earlier than that, because you might not get the maximal indel rate. And throughout this process, again, maintain representation at greater than 500 cells per guide. And for most screens, I would recommend starting with 2 bioreps, and then if the data looks good, then do another 2 bioreps. It really helps clean up the data. For an activation screen, the first couple of steps are the same. You put in the activator components at an MOI of less than 0.7, then put in the guide RNA library. And for activation, because this acts much quicker, you can start the screen as early as 4 days after library transduction. So general considerations when you're applying your screening selection, obviously this really varies from screen to screen, but basically you want to choose screening parameters that maximize the difference between the experimental and control conditions. Typically the way people do this is that you know a couple of genes are sort of positive controls, a couple of genes are negative controls, and these 2 sets of genes will help you set, like, for example, what drug concentration you're using, or like, other parameters that affect your screen during the selection process. For drug selection, we usually use the IC50 value of the drug, because that value avoids over-selecting and under-selecting your cells during the screening process. Again, it's very important to maintain the coverage of over 500 cells per guide in your library throughout the process. If it's the first time setting up a screen, you don't know how long to run the screening selection for, I would recommend just collecting multiple time points, because if you're working with dividing cells, chances are you'll have a lot of extra cells if you're just maintaining 50 million cells throughout the screen. So what you can do is at week 1, freeze down the cell pellets at -80 and just keep them there, and harvest them if you think those will be useful. And because like freezing down the cell pellet really is not too much work, but redoing an entire screen is a lot of work. And throughout the screen, it's also very important to remain very consistent. So for screening, we won't change batches of FBS, for example, because even things like that, for sensitive screens, that makes a huge difference to your screening result. Okay, then at the end of the screen, we will harvest the cells for screening analysis. In the protocol, we use Rigor, which was originally developed for analyzing shRNA screens to identify candidate genes. The way Rigor and many other screening analysis tools work is that you basically normalize the experimental guide counts to the control based on NGS counts. Then you rank all of your guides based on the ratio of experimental guide count to control. And then for each gene, you look at the ranking of all the guides that target that gene, and then figure out the statistical significance of whether that gene is significantly enriched or depleted. Right now, I think the best tools that are out there is probably Magic, developed by another group. Magic is a lot more updated, and I think besides Magic, there's also STAR, which is very useful for screening analysis. And then after you get all the gene rankings from the screening analysis, you can take either the average of the rankings or take the overlap of the top genes to figure out which ones you want to follow up on. So the last step of the screen is a validation process. The validation arguably is probably the most important part of screening, and it's going to be the part of the process that takes the longest time. The screen is very noisy, and at the end of the day, you get a ranked list of genes, but you really don't know if each gene truly confers your phenotype. Obviously, if you set up the screen very well, and there are a lot of genes that are involved in your screening phenotype, most of the top 10 will be genes that are involved in your phenotype. But for a lot of screens, the phenotype is very mild, and not all of the genes that are enriched will be the ones that confer your phenotype. So you want to go back and introduce the target guide RNAs for each gene individually to make sure that the phenotype is what you would expect from the screen. So for the first part of this, what you will then do is pick the genes that you want to validate, pick all the guides that, that target that particular gene in the screen, and clone each of them individually into the vector that you use for your screen, package it into lentivirus, and then transduce and select. Same way as you did your screen, except now all the guides are individually packaged. So you know that if there is a phenotype, your guide was the one that conferred the phenotype. So for a knockout screen, after all of this, we usually will extract genomic DNA from the validation cell lines. And then the protocol, we use 2 rounds of PCR for the target site with Illumina adapters for NGS. And the reason why we do 2 rounds of PCR for this process is because usually you're validating say at least 30 to 40 different target sites. And so you want to have PCR primers that are very versatile and can append different barcodes to different target sites. And so we recommend the 2-round PCR for this process. And for the screening phenotype, you need to, again, check that these guides do confer the screening phenotype. Assess the indels, and then a lot of times what people will do is verify by Western blot that the protein of interest was actually knocked down. For activation, what we provide is a homebrew protocol for rapid extraction of RNA and reverse transcription. And this protocol doesn't involve the column. You basically take a plate of 96-well, plate of cells, typically 96 wells, and then you add the lysis buffer to it, mix up and down a couple of times, and then transfer this mix into the RT reaction. And this is very similar to, this is basically a reverse engineered version of the commercial cells to CT protocol, which costs about, I think, a couple $1,000s per 96 well plate. And in contrast, our protocol costs about maybe 1 or 2% of that. So it's very cheap, very easy to use, and I would recommend if you're not doing, even if you're not doing screening, at least take a look at the RNA extraction and RT protocol that we provide. That is very cheap and very easy to do. And then after this, we recommend doing the TABMan qPCR. The TABMan qPCR is preferable to the Cyber qPCR because there's a 3rd primer probe that basically releases fluorophores when the polymerase goes through it. And so it provides an added layer of specificity to the primers. Then again, verify the screening phenotype and make sure that the guide RNA actually activated the gene of interest by assessing the mRNA and by assessing the protein by Western blot. So finally, the most important screening considerations is, again, I've repeated this throughout the screen, but I can't emphasize enough how important it is to maintain the screening coverage of over 500 cells or molecules or whatever throughout the screen. And it's very important to include controls such as non-targeting sgRNAs or sgRNAs that target genes known to affect your phenotype. And then it's also very important to include control conditions that did not have your screening selection, so you know that what the distribution of guides look like without applying the screening selection. And then throughout the screen, remain consistent, use the same protocols for screening and validation so that everything checks out. And finally, I'd like to thank all the people who helped me put together this protocol, everyone from the Joung lab. And thank you very much, and I'll take questions now.
Hey, Julia. I have a question about validating the guides. So you said you use the guides you're using in the screen. Why is it possible to use independent guides targeting the same gene?
So some people would do that too, in addition, to make sure that the guides, basically the phenotype is not due to some off-target from some guide in the library. So that's also possible. I usually will start at least with the guides from the library so that you know that there should be a phenotype, right? And then if you're worried about off-targets, usually that's not a huge concern if you're using multiple guides. But if you're really concerned about off-targets, you can always use independent guides.
I had a question in respect to the activation screens or the inactivation screens, so the knockout screens. In terms of where you design your guide RNA closer to the transcription start site, or for example, enzymatic domains, or a mix of both, would you be able to comment on which ones are more effective?
So for knockouts, there's actually a paper showing that if you target the functional domains of the protein, for instance, kinases, I think they did it for kinases, you get a much better screening result. In our genome-scale library designs, we didn't do that because for most proteins, you don't actually know what the functional domains are. So what we do is we knock out at the 5' constitutive exon and hope that it generates a missense, a frame shift in the protein, and that knocks out the entire area. For activation, what we found is that if you target the 200 base pairs upstream of the annotated transcriptional start site, for all the genes that we've tested, this leads to a very robust transcriptional activation of the target gene. And so that's how we designed our library.
Finally here. One question regarding the targeted knockout, not knockout the whole gene, but particular domain. So is there any study that they can modify protein-protein interactions, because we know a lot about protein interacting domains? And are there any studies that particularly targeted that and see if we can efficiently modify protein interactions instead of knocking out the whole gene, but more targeted in that aspect?
So in a screen, not that I've heard of, but I'm sure in basically for single genes, people have done that to specifically assay a particular, basically a particular interaction versus frame shifting the entire protein.
Okay. So thank you, Julia. And Julia will be available during the breakout session for questions. And Agilent will also be hosting a breakout session. And they'll be talking about ultra-high quality custom guide RNA libraries for CRISPR-based functional genomics. And you may be able to get some other tips from them about designing libraries for screens. So I think we'll... So we'll move on to our last talk for this session. Which is kind of an introduction to RNA targeting systems. So Max Kellner, who is a visiting master's student in our lab from the University of Vienna, has been working a lot with some of these enzymes, and he will give you guys an overview of these enzymes and some of the tools that have been built using them.
So hi, my name is Max, and I'm a visiting master's student, as Rai pointed out. I'm going to switch gears now, and instead of talking about DNA targeting enzymes, I'm talking about RNA targeting enzymes, and specifically CRISPR-Cas13, which kind of has been demonstrated to target RNA for the last couple of years, 2 years actually. So for my today's talk, I would like to talk about the what, why, and how of RNA targeting using Cas13. What in terms of introduction to CRISPR-Cas13 biology, in some in vivo and in vitro applications to tell you why we think RNA targeting enzymes could accelerate RNA biology research. And finally, some design principles if you want to implement this during your experiments. So this is kind of a summary to talk about the class 2 effectors. CRISPR effectors, we have heard about Cas9 and Cas12a. Cas12a also CPF1, so these were renamed to Cas nomenclature. So Cas12a, as I said, is CPF1. So instead of targeting DNA, like Cas9 and Cas12a do, Cas13 targets single-strand RNA. And it does not use the conventional rough COH and H domain like the DNA targeting enzymes, but it uses heptN domains, and what that is, I will explain in a little bit. But there are also similarities to other Cas2 effector enzymes, including the guide RNA structure. So you can see that it's actually very similar to CPF1 CRISPR RNA, having a direct repeat sequence, a spacer sequence that's complementary to RNA. Instead of using a PAM, so protospacer adjacent motif, like Cas9 and Cas12, it actually, at least in vivo, does not use such a motif. In vitro and in bacterial cells, it was found that something like a protospacer flanking site exists, so a single nucleotide, that can affect targeting efficiency. Yeah. So these heptene domains are essentially conserved among Cas13 enzymes, and they're also found in other RNA targeting enzymes, even in the ones that are found in human cells like RNA-seq. And these heptene domains are marked by two conserved residues, arginine and histidine. And traditionally, heptene domain-containing proteins have a methylene-independent catalytic RNA degradation step by the fact that they dimerize to form a cleavage interface. Cas13 kind of gets around the dimerization by having two of these domains implemented in one single factor complex. But Cas13A is not the only CRISPR enzymes that contains these heptene domains. There are also other ones found in the CRISPR type 3 system, namely CSX1 and CSM6. So this suggested in the beginning that because Cas13 has heptene domains, it probably is an RNA targeting enzyme. This is kind of to show you the CRISPR type 6 diversity. So in the last year, there have been numerous papers showing that there are different subtypes of the type 6 family, starting with CRISPR-Cas13A, which was first described biochemically in 2016, and then followed by Cas13B, C, and most recently D. And the reason why I show this slide is to show you the diversity of the loci. So as CRISPR systems, they all have a CRISPR array, but you can also see other features like accessory proteins within that array, namely Cas1 and Cas2, which are known for space acquisition integration, which are not found in other type 6 subtypes. But then also interesting accessory proteins found in these loci, namely, for example, CSX28 and CSX27 found in CRISPR type 6b, 1 and B2 systems, but also Cas13D has accessory domains. And these were found in vivo to at least modulate activity. But one very interesting observation recently is found that Cas13D family are actually much smaller than other RNA targeting enzymes like Cas13A and B. As I said, with these heptene domains, Cas13 is an RNA targeting enzyme, and this is shown here that Cas13 operates in an RNA-guided manner. So without guide, you don't get cleavage. And it's also, interestingly, to other heptene domain-containing proteins, methylene independent. So addition of EDTA kind of kills in vitro RNA degradation. And to the difference to RNAi or shRNAs or microRNAs, which usually have a single cut site. So if you do such a gel, you usually get like 2 fragments because it cuts once. Cas13 seems to cleave the transcript across this line, something that is either a native biochemical property or a collateral activity, which is something that Cas13 has, and I will come to in a minute. This Cas13 system, as I said, also has a weird, interesting property called collateral RNase activity, which essentially means that when Cas13 loaded with a guide recognizes a target that's complementary to its CRISPR RNA, it unleashes RNase activity to a bystander RNA, an RNA that is not specific or complementary to the CRISPR RNA. In this case, it's labeled as collateral RNA. So this gel-based readout shows that if you have a labeled bystander RNA, collateral RNA, upon addition of CRISPR RNA and target, you kind of see cleavage of this collateral RNA, something that has been turned into a nucleic acid detection tool, and I will finish my talk actually with this application. To summarize Cas13 biology, Cas13 is a CRISPR type 6 system. It has a dual RNase activity where one activity processes the CRISPR RNA, the pre-CRISPR RNA. So this allows you to, for example, for in vivo applications, have a CRISPR array and have Cas13 to process these in the individual spaces. This catalytic activity is independent from its RNase activity, endoRNase activity, which can be specific, so cis, acting on its RNA molecule that it's bound to, or in trans, after activation of a complementary RNA. So why we want to use Cas13? There are numerous applications that you can think of for in vivo studies. This is from a review from 2011, where people looked into other RNA binding proteins, how they can be used to kind of engineer the transcriptome. For example, you can imagine that you want to visualize RNA. RNA visualization has typically been done by inserting transgene sequences, like MS2 sequences, which are then recognized by code proteins. But this is kind of an engineering approach that is not amenable to endogenous genes. So having RNA binding proteins allows you to kind of visualize these endogenous genes by fusing a fluorescent protein onto it. You can also imagine that you would, for example, like to understand how localization of an RNA affects its function. So by fusing effector domains to your RNA binding protein, you can kind of alter localization. As Cas13 and other enzymes are RNA targeting enzymes, you can essentially degrade RNA in a targeted manner. But you can also play around on more sophisticated levels, for example, modulating translation by enhancing it, fusing initiation factors onto it, or repressing it. And you can also modulate splicing by just simply designing binding proteins, or in our case, Cas13, that targets splicing sites. So you can modulate splicing by including or excluding certain sequences. So the first application we'd like to talk about is RNA knockdown in eukaryotic and prokaryotic cells. This is kind of an example from the first demonstration of this in mammalian cells, where we have a Cas13 expression plasmid, where the CRISPR-RNA is like with Cas9 driven by U6 promoter. You have nuclearization sequences attached to Cas13. In addition to that, you have a CRISPR-RNA expression plasmid. And for this particular assay, we also have a reporter plasmid that has 2 luciferase variants onto it, one of which is targeted by the CRISPR-RNA that's found in the CRISPR-RNA plasmid, which allows you, after transfection, to essentially read out Cas13 activity by having relative luminescence, bioluminescence activity. This is kind of to show that among the Cas13 variants tested, all of them have kind of robust knockdown activity, shown on the left, with Cas13 in our hands being more potent in RNA knockdown. What we have here are 2 guides targeting luciferase, and we plot here the guide normalized values for this luciferase. And you can see that, for example, Cas13b6 and b11 are very potent in knockdown. Surprisingly, is that Cas13 in vivo does not display a collateral RNase activity. So if you do RNA-seq and look at differentially expressed genes and compare this to conventional knockdown strategies like RNAi, you actually don't find off targets, which is surprising given that in vitro you have this collateral RNase activity. Next, I would like to talk about applications for RNA imaging using dead Cas13. So you kind of take Cas13 and mutate hapten domains, which are the RNA targeting domains, and then cleave RNA. So if you mutate those, you kind of use RNA kind of for all binding applications, including imaging. And the first application done by Jonathan Omar in our lab, where they used Cas13, fused GMS-GFP for its folding property, but then also a zinc finger and a crab domain, which essentially allows this to be a negative feedback. Because, for example, if you have Cas13 fused to a fluorescent protein and constitutively expressed, this kind of results in a strong fluorescence independent, you can imagine, for its ability to target RNA. So what they used is they fused this repressor domain together with a zinc finger onto it, so that when the RNA that is targeted is not present, it should repress its own transcription and not being translocated into the cytoplasma. This is kind of one of the paper figures where they showed beta-actin targeting guides, 2 targeting guides, and a non-targeting guide. And you can hopefully appreciate the subcellular localization that is distinct among those. The middle lane is a staining for stress granule formation. So these cells were treated with sodium arsenide to allow for visualization of stress granules where RNA molecules are trapped. And these RNA molecules are targeted by Cas13 loaded with the beta-actin guide. And you can see that in the overlay image of staining. Linyi mentioned this, but CRISPR-Cas13 can also be used for RNA editing. So maybe you've heard about Cas9 DNA-based editing, where Cas9, dead Cas9, was fused with a domain that deaminates bases. Similarly, we have engineered Cas13 fused to ADAR, or the deaminase stain of ADAR2, which allows us now to do targeted base editing on an RNA level. And the picture below kind of shows the architecture of such an interactions, where you have the guide. And within that guide, you place a mismatch that's opposite of the A you'd like to deaminate, because this is an adenosine to inosine deamination event. And this mismatch allows a hyperactive variant of ADAR to specifically edit this site. So unlike DNA-based editors, which usually have like a window, we can specifically alter that one site, which allows for base conversion on the RNA level, something which could be reversible instead of permanent DNA changes. But I'm not going into more details about this, because Omar in our lab will have a talk about this tomorrow. And to finish on the in vivo applications, it was also shown that Cas13, dead Cas13, can be used for modulating splicing by targeting certain sequences found in exons like donor and splicing acceptor sites. And Silvana will talk about this tomorrow. So again, I'm also not going to detail into that. So these are very exciting applications for in vivo targeting. So some design principles. It's actually very cheap, very easy, because all our Cas14 or demonstrated Cas14 expression plasmid have been deposited on that gene. So what you can do is simply type in Cas14 on that gene and select your expression plasmid of interest. The only thing you essentially need to do is design CRISPR-RNA spaces as DNA oligos, hybridize them together, and Golden Gate clone them into our CRISPR-RNA expression plasmid, which you can also get from ad genes. Then you transform this into E. coli, harvest the plasmid, and it's ready for transfection. So for transfection, you essentially deliver these agents and harvest RNA about 48 hours post-transfection, and then you read out RNA targeting efficiency by either qPCR, report of fluorescence or bioluminescence, as I've shown with the luciferase vector, or RNA-seq, which allows us to actually look at the sensitivity and specificity of RNA targeting. And now I'd like to home one more time into this collateral RNase activity, which seems to be a case in vitro, but in vivo we have not found evidence for that. But in vitro, at least, it's a very useful application. So we kind of use a Cas14 not only for transcriptome engineering in living cells, but also for detecting. And you can ask yourself, okay, why do we want to do that? It's because there are several applications which require actually on-site diagnostic platforms where you don't have sophisticated equipment, including like a thermocycler or a fluorescent plate reader of any sort. So this can be useful for viral detection or discrimination between bacteria, but also because of its sensitivity, detecting rare events like DNA mutations found in cell-free DNA. So our lab has turned this into this Cas14 detection into a diagnostic tool by combining it with pre-amplification, where your starting material is either DNA or RNA, which is then isothermally amplified with RPA, recombinant polymerase amplification reactions, where the main difference is that either it has a reverse transcriptase added or not, so you can actually have RNA or DNA starting mount, which converts that into double-stranded DNA, having a T7 promoter introduced by the primers, which is then turned again into an RNA molecule by in vitro transcription, and then you leverage Cas14's collateral activity to kind of recognize that specific RNA molecule, but because it undergoes this conformational change, it allows you to collaterally cleave a reporter, which unlike Tachman probes is not specific to a target sequence, but can be a uniform reporter molecules used for all your applications. So the variable tool in this application is essentially your guide RNA. So Cas14 by itself is not very sensitive, but if you couple that with pre-amplification, you can get down to single molecule per microliter levels, or more recently, 2 molecules per milliliter, something that is actually the case for Zika infections. And as I said, it's not limited to RNA. You can also use DNA as a starting amount, shown here, by essentially having the same sequence that is targeted. One form is RNA, one form is DNA. So in both cases, you get very high sensitivity. To kind of get around the requirement for a fluorescence signal, our lab has turned this into a low-cost portable and colorimetric platform by kind of changing the design of a reporter and using lateral flow technologies, similar to a pregnancy-based test, where an uncleaved reporter is sequestered on the strip under the first line, control line, because it has an intact biotin tag, which sequesters the whole reporter under this first line. But upon cleavage of this reporter, you kind of separate these two parts together, where one FAM is recognized by an anti-FAM antibody and travels onto that second line, the antibody capture line, which allows you to read that out similar to a pregnancy-based test. And this shows also very high sensitivity, down to 10 to 100 molecules per microliter. And we're currently optimizing it to be more sensitive. Some design principle for Sherlock, it's similar to in vivo applications. So you design your CRISPR-RNA having a 5 or 3' repeat. It's an important consideration because these different Cas14 enzymes have the repeat either on their 5 or 3' end. We currently use a 20 nucleotide CRISPR-RNA spacer that's complementary to your RNA target. And if you design this as a DNA ultramer and want to produce your own guide instead of ordering an already made RNA targeting, you have to include a T7 promoter, or this as reverse complement, so your RNA is made in the right orientation. You also need to design RPA primers, which are similar to PCR primers, with the difference that they are a bit longer in size. And because you want to turn that DNA molecule into an RNA molecule, you also have to include a T7 promoter into its forward primer or reverse primer, depending on which strand you want to target, which then allows you to kind of turn that into an RNA and detect it. And finally, you have to order all Sherlock reagents, the RPA kit, which is from TISDX, which is available as RTRP or RPA kit. Then you have to make essentially Cas14, because unfortunately, there's no commercially available Cas14 protein, as far as I know. But all these expression plasmids that we and others have used in papers are found on ad genes. And these are sumo-tagged expression vectors, so the purification protocol is fairly easy, and these are highlighted in our papers. So currently you have to make Cas14 yourself, but this is actually not a very complex task if you're familiar with protein production. You also need to buy T7 RNA polymerase, an essential component of this in vitro transcription reaction finally, and your fluorescence or colorimetric reporter for the detection part. So basically, if you want to implement Cas14 now in your research, or you're kind of lost in the CRISPR diversity and don't really know should I use Cas14 A, B, C, or D, or anything about your RNA targeting experiment, just come see us after my talk, essentially after the coffee break, because we will have breakout sessions. But this is not only true for RNA targeting, but also for other CRISPR applications. So just come see us after the coffee break, and we have breakout sessions. With this, I'd like to thank the Joung lab, Jonathan Omar, who have trained me over the last year to be a kind of, at least, lab expert in Cas14 biology, Rhiannon and other GE co-organizers, and you all for listening and participating in this workshop. Questions?
Apparently, I'm sitting in the worst place. Quick question about the imaging ones for visualizing RNA, mRNA. I was wondering, is it a quantitative or qualitative? Can we actually measure the amount of RNA, and with what kind of accuracy can we use that in order to, let's say, particularly we're interested in one particular RNA, can we trace it live to see like if its expression is changes in response to particular event, and B, like how much like range of dynamic range can be measured?
Yeah, so it's a good question. So in the paper published in Nature last year, we have essentially demonstrated in situ RNA detection imaging, but not on a single molecule level. So to answer your question whether it's quantitative or not, we have not explored single molecule amplification, single molecule imaging technologies. But yes, you can image this in life, and we have done that in the paper, and you can essentially highlight this. So we observe these stress granular formations in living cells, something that is useful because now you can target actually endogenous genes or in vivo tissue. Yeah. Yeah. Yeah, yeah. Yeah, so that's an interesting question because there are many diagnostic platforms which require you to have, for example, very pure starting material. In a paper published about 2 weeks ago, which we co-authored, we actually showed that you can use urine for direct detection. So you essentially heat treat urine to kind of deactivate endogenous RNases, and you can then use the sample directly. We've also played around with direct crude extracts from genetically modified plants, so you can do direct GMO detection by just simply grinding them in a very easy lysis buffer. So it seems to be a very robust technology in terms of contaminant sensitivity.
Hello, and we have seen that the Cas13 system can be used for RNA editing.
Yes.
And one of the applications being the localization of an mRNA can be actually changed.
Yes.
So let's assume that the mRNA has a different signaling sequence, let's say a mitochondrial localization signal.
Yes.
Can that be replaced with a nuclear localization signal using Cas13?
So you're essentially saying that you want to use RNA editing to convert this mitochondrial signal in any other sequence. Or you would just like to target it by Cas14, which has an effector that then delocalizes or relocalizes it to a different place. Is that the question?
I need it to be specific to the nucleus.
Yeah, so what you can do is essentially target this RNA by having a nuclear localization signal on your RNA targeting enzyme which is Cas13, which makes it a very nice tool for also targeting or imaging nuclear localized genes, something that usually is very hard to achieve with RNAi technology. So yeah, you can redirect the localization of RNA molecules by having the localization tag present on your Cas13 enzyme.
Does that mean that the mitochondrial localization is being is redundant, or it still exists in the RNA along with the NLS?
Yeah, so I cannot give you a detailed answer, but maybe we can talk about this after this talk. So I don't think we have explored this in detail, but it should be possible.
Okay, thank you.
All right.
Okay, so, all right, thank you, Max. And thank you to all of our speakers in this first session. Yeah. So before we break for coffee, I just wanted to bring your attention again to this. So we'll do like a 15 or 20-minute coffee break, and then after that, we'll have breakout sessions. So we have 4 different sessions. These will run concurrently. You're free to move from one session to another. In the auditorium here, kind of at the front, we'll have experts on Cas9, and at the back, we'll have experts on Cas13 for any kind of RNA targeting applications. And then in the lobby itself, Lin Wu is here. She's the director of the genome modification facility at Harvard, and she will be speaking about making mouse models using CRISPR, and she'll be out in the lobby. And if you go through the lobby to the very end, there's the Olympus boardroom, and Agilent will be in there, and it'll be sort of a general screening session. So Julia will be in there, who spoke earlier, and Agilent and some other people will be in there to kind of talk about screening applications. And then through the lobby and upstairs will be Benchling, and they have a number of CRISPR guide design tools and just general electronic lab software that is really neat, and a lot of people in our lab use it. So I think they will also hopefully have some helpful things to say to you guys. So enjoy some coffee, and there's some snacks out there, and then we'll see some of you back here in the auditorium for breakout sessions. And we'll meet back in the auditorium at 4 o'clock for the keynote speak.
All right, good afternoon. Welcome to Genome Engineering 6.0. We'll have guests from all 4 corners of the US and from many other countries around the world. So thank you so much for coming here. And I hope the next day and a half will be a series of very exciting and stimulating conversations and learning experiences. So if you didn't have your questions answered so far, a lot of us who will be wearing these gray t-shirts with the Cas9 schematic on the front will be around. So feel free to find any of us during the next day and a half, and we're happy to answer any questions. And of course, after the symposium, feel free to reach out to us either directly or via the Google forum, and we'll try our best to answer as much as possible. And then after this session, we're going to have a poster session, and there will be beer and drinks and some food. So hopefully that will help lubricate further discussion around genome engineering. Anyway, but here, I think we're all here for the main program for this afternoon, which is the keynote by Eugene Koonin. For those of you who may not be familiar with Dr. Koonin, Eugene is one of the premier scientists at the National Institute of Health in the National Center for Biotechnology Information. He has been working on CRISPR before there was even CRISPR, and had put forth many of the fundamental understandings and functional descriptions of what CRISPR may be doing. And since then, Eugene has been pioneering the investigation of CRISPR diversity. A lot of our understanding of how CRISPR systems are organized really comes from the work that Eugene and his colleagues at NIH have been doing. And this is only a small fraction of what Eugene has done. He's published over 800 papers. And I think of those 800 papers, CRISPR is only a small fraction. And there are many, many other really extremely fascinating aspects of evolutionary biology, microbiology, even evolution of eukaryotic biology that Eugene has really sort of, it's got his DNA all over those different topics. Anyway, without further ado, let's welcome Eugene to teach us more about CRISPR and broader evolution of biological systems.
Good afternoon. It's a great pleasure. It's a great honor to speak once again at this wonderful workshop. As I told Fang before, it has really, I'm speaking here for the 4th time in a row, and it has really become one of the hallmarks of the year for me. First of all, I want to thank the organizers very much, and most particularly Fang, for putting this all together and making this possible. This is what I'm pleased to say every year here. This year, however, there is something very special. I want to congratulate Fang on his election to the National Academy of Sciences just a few days ago. I think he richly deserves that, and a reach and applause from all of us right now. I'm going to talk to you about expanding the CRISPR horizon, the CRISPR universe, so to speak, towards deeper, at some point eventually, comprehensive understanding of what CRISPR are, what are the entirety of genes that are functionally linked to the CRISPR systems, and more importantly, of biological processes in microbes, to which CRISPR make contribution, as we shall hopefully see towards the end of this talk, far beyond straightforward defense functions. I have to apologize for the error on this slide here. I have a previous meeting indicated here. For anyone who might have been present at that meeting, and I know several people were there, the overlap is minimal. Okay. This slide shows you the layout, or modular layout of the CRISPR-Cas system. Somewhat arbitrary, but I think not entirely rooted in the functions of the products of the Cas genes classification, which divides the Cas proteins between distinct modules that are involved in adaptation, acquisition of spacers, which makes CRISPR the adoptive immunity system it is, interference, which is the actual recognition and disruption inactivation of the target genomes, which makes the CRISPR the efficient defense system it is, and expression and pre-CRISPR RNA processing, which is a necessary intermediate step. But beyond and apart from this, there is this ancillary module of the genes that are often associated with CRISPR-Cas systems, but whose functions, as we understand them, do not immediately fall into one of these other modules. And it is into this ancillary or accessory module where I want to venture today with a variety of analysis that we have done and that I will try to describe to you quickly enough. But I want to start in a somewhat more traditional manner and gradually build up to go into these other dimensions of CRISPR. So the slide that comes from a review review paper that we published with Fang and Kira Makarova about a year ago, and shows you the latest classification of the CRISPR-Cas system. I will not, it's not my goal today to describe this to you in detail, but just a couple of points. At a deepest level, the CRISPR-Cas systems are divided into two classes, class 1 and class 2, where the difference lies in the organization of the effector modules. That is, those modules that are concerned with functions downstream of adaptation, processing of CRISPR-RNA and interference. So in class 1, these are very elaborate complexes of multiple Cas proteins with effector modules, whereas in class 2, it is at least at face value on the surface much simpler. The all effector functions reside in a single large protein, such as, for instance, Cas9, which makes the systems of class 2 so attractive and in some cases so efficient genome editing and genome engineering tools. So most of my talks at the previous editions of this workshop were dedicated in one form or another to the discovery and characterization of novel class 2 CRISPR-Cas systems, in particular discovery of several new subtypes of type 5 that shed light on the evolution of the effector complex, and of type 6 that is the first and so far only case of CRISPR-Cas system dedicated to recognition and cleavage of RNA, and that in the able hands of Fang's lab and some others have now become very, very useful tools of molecular biology. So I want to expand on this even more, even today. So just very recently, we managed to further expand subtype 6D through the efforts of the alumni of Fang's lab, now at the Arbor Technologies company. Dave Scott, Winston Yan, and Dave Chang. I believe Dave and Winston are here now, so good. This was a discovery and characterization of the novel new subtype of type 6 systems, which we denote Cas 13D, which was simultaneously and independently studied by other alumni of Fang's lab, Philip Su and Silvana Kornerman in San Diego, and which is very interesting in that it includes the smallest, the shortest proteins among all type 6 varieties, which obviously about 20% smaller than others, which makes them obvious good candidates, attractive candidates for new molecular biological tools. But there is something else that I want to emphasize as a transition to my main subject today. That other aspect is that these Cas13D effector proteins are associated with a special kind of accessory proteins, which I will describe in some detail. So phylogenetically, Cas13D is a sister group of now well-characterized Cas13A, and you cannot see much in the alignment on this slide, but what you can see, I think, is the diversity of these proteins, which is an intrinsic feature of many Cas proteins, but Cas13 in particular, beyond the two hep-N domains. You really do not, which are the active ribonuclease domains, you really cannot see much conservation even within this group, let alone between. The Cas13D subtype 6D in itself is fairly diversified, found in a variety of diverse bacteria as well as in metagenomes, and the notable feature of the locus organization here is that almost always the Cas13D gene is accompanied by another gene which encodes, encodes the protein containing the so-called WIYL domain, or will domain, as we pronounce for simplicity, which is not particularly well characterized, but is at least predicted to be a nucleotide-binding signaling domain. And in these experiments reported in this paper, Winston and others have shown that addition of the will domain proteins significantly, by about an order of magnitude, increases the efficiency of interference by the Cas13D protein. So in addition to the attractiveness as potential tools, the study of these systems reveals a novel type of regulation of CRISPR activity. Incidentally, when we look back at the literature, what we notice is that these will domain containing proteins can also regulate certain type 1 systems. In this paper by Wolfgang Hess and Koberkers, it has been the stimulatory effect has been shown on the activity of type 1 systems, and it has been proposed that this protein is a transcriptional activator. The results in this paper suggest this is probably not the case. This is done through direct interaction and ligands which remain to be identified. So that was a brief introduction and connection to my previous presentations here, where we discovered new types of effectors of CRISPR-Cas systems. Now I want to focus more on the ancillary or accessory proteins. And I want to begin with this, by now I think rather well known to everyone who is interested in CRISPR, but I think spectacular case in point. Type 3 CRISPR-Cas systems, all of them, contain the Cas10 protein, which is the large subunit of the effector complex. As I mentioned a few moments ago, in class 1 CRISPR-Cas systems, including type 3, the effector complex are elaborate machineries made of multiple distinct Cas proteins. And the largest of them, the large subunit, is the Cas10 protein. But apart from the structural, my colleagues and I predicted quite many years ago, as Frank said in his introduction before CRISPR, that these proteins contained all the signatures of catalytically active nucleotide polymerases or cyclases, and therefore must possess this activity. For many years, this prediction has not been tested and became a source of a bit of an embarrassment, if I may say so. However, things changed about a year ago. Two laboratories of Martin Ginnik in Zurich and Virgis Siksnis in Vilnius simultaneously discovered the activity of the polymerase cyclase domain of Cas10 protein and the role of this activity in the functions of type 3 CRISPR-Cas systems. So what happens is this. Upon target recognition by the effector complex, the conformation of Cas10 changes and the polymerase cyclase domain gets activated and starts churning oligoadenylate, starts oligoadenylate synthesis. These oligoadenylate become ligands for the so-called CARF domain that is present in another in an accessory protein, which is found not in all, but in the majority of type 3 systems and known variously as CSN6 or CSX1, and consists of this nucleotide binding CARF domain, which means CRISPR-associated Rossmann fold, which was another of our computational predictions that until these works have been completed remained untested, and another effector domain, which most often represents a HEP-N domain, ribonuclease homologous to that in type 6 CRISPR-Cas systems, and sometimes other nucleases. So it has been shown in these studies that the CARF domain binds these oligoadenylates produced by Cas10, and this binding changes the conformation of this protein and activates the HEP-N domain that then starts non-specific DNA degradation, not of the target RNA, but of all the RNAs around, potentially, as we strongly suspect, connecting CRISPR-Cas response to programmed cell death in the infected organisms. In any case, I think it's a very remarkable story that shows to us that these accessory proteins, the CRISPR-Cas systems, can be involved in whole pathways of signal transduction, whose role probably goes far beyond CRISPR. So continuing along these lines of discovering the functions of CRISPR ancillary or accessory proteins, here is a protein whose function is rather poorly understood, denoted CSN2, which is present primarily in type 2A CRISPR-Cas system. So here we momentarily move back to class 2. This protein is known to inhibit the non-homologous end joining repair by type 2 CRISPR-Cas system. What is the biological role of this inhibition is still rather unclear. What we found to our great surprise with Kira Makarova in still unpublished analysis is that this CSN2 protein is actually an inactivated P-loop ATPase. So in the ancestral forms of this protein that can be reconstructed by the analysis of phylogenetic tree, apparently all the catalytic motifs required for ATP hydrolysis have been present, and the protein performed some kind of ATP-dependent, energy-dependent, ATP-dependent function here, which probably still can be elucidated in those sort of relics of ancestral systems where the proteins remains, according to our prediction, active. In the great majority of the CRISPR-Cas systems, it has been, the ATPase activity has been degraded, lost, and the protein assumed purely structural function. So all these observations prompted us to go around this business systematically, to derive methodology, to derive and apply methodology to systematically identify ancillary genes in CRISPR-Cas loci, try to predict their functions in the CRISPR-Cas response and beyond, and distinguish functional associations from spurious ones. So in order to do so, we did what we do always, namely develop a computational pipeline, a dedicated computational pipeline. This pipeline, we denoted the feature we were after. We denoted it CRISPRicity. And CRISPRicity is supposed to mean the association, the quantifiable association of a particular gene with CRISPR-Cas systems. I shall note that this approach is quite generalizable and general. By doing so, you can explore all kinds of functional connections in microbial genomes. Of course, speaking very simply, this is all based on the now well-known fact that has been established by Jacques Monod and Francois Jacob almost 60 years ago, that microbial genes very often are associated, functionally linked microbial genes often form operons, string of co-expressed genes on the chromosome. And, as it has become clear later, the operons are highly variable. So you really gain a lot of information by comparing the operon organization in different bacterial genome. Specifically when it comes to CRISPRicity, what we do in using other previously developed computational pipelines that I don't describe here is comprehensive identification of CRISPR loci in a prokaryotic genomic database. And we do it by sort of three-pronged approach. We detect CRISPR arrays using the appropriate computational methods. We detect adaptation modules, Cas1 and Cas2. And we detect effector modules. Because while many CRISPR-Cas systems have it all, there are quite a few that actually have only one or two of these elements. So we applied the previously developed methods to identify each of these elements, and then enumerated all the proteins in the respective genomic neighborhoods from all genomes that contain at least one of these elements. Then we clustered all the proteins to work with gene families rather than with protein families rather than with individual proteins. And then we determined the strength of the association. The strength of the association is a very simple thing. It's simply the number of occurrence of a given gene in the vicinity of CRISPR loci, normalized by the total number of occurrence, which shows you how specific the association of a given gene with the CRISPR-Cas systems is. But then you have to do extra analysis in the multi-dimensional space. I will not go into this in much detail, but you have to add factor in the abundance of genes, because it's very clear that if you have the number 1 here, you have only one instance in CRISPR vicinity, and number 2 here, 2 occurrences of a gene altogether, it will formally give you high CRISPRicity, strong association, but obviously will mean very little. So we have to factor that in, the abundance and the actual distance from these elements, because the closer the better, so to speak, the stronger the association. And you have to isolate the domain of the parameter space, which contains proteins close with properties close to the known Cas genes. And these are our candidates that we afterwards explored in great detail by case-by-case analysis with various computational methods. So what comes out of it is this very simplistic pie chart. What it tells you is, first of all, that there are quite a few highly divergent versions of known Cas genes, such as, for instance, Cas5 and Cas7, so-called ramp domain containing proteins, which are known to evolve very fast, to be very highly divergent, and we identified many additional families, which are important but unspectacular findings. But also, we detected quite many new proteins that did not belong to new, of course, in this context, they are not necessarily completely new, as we shall see, that appear to be strongly associated with CRISPR-Cas systems, but have not been described as potential Cas proteins before. And we also have to indicate that this is not a rigorous procedure. We don't know, to be completely honest, how to develop any kind of rigorous statistics for this type of analysis. So case-by-case examination shows that about half of the high CRISPRicity genes, for various reasons, because of their predicted functions, because of lack of evolutionary conservation, whatever, are unlikely to be CRISPR-associated. That said, let's concentrate on the positive. The crop, I think, is fairly good. There are about 100 new or potential new CRISPR-associated genes and encoding proteins. So immediately we classify these novel potential CRISPR-associated genes by CRISPR types and subtypes, and we immediately see something quite interesting, namely that a substantial majority of these new candidates for CRISPR association are found in type 3 CRISPR-Cas loci. There seems to be, in terms of the richness of biological functions, something special about type 3. We shall talk about it more. So, of course, we have outstanding questions here. How do we decide whether the association is real or alternatively, a particular gene is just part of a defense island. Microbial genomes contain defense islands, parts that are enriched with different defense systems, not necessarily directly functionally linked, sort of junkyards of microbial genomes where you can insert all sorts of things, and so defense systems accumulate. So how do we differentiate between these different possibilities, and how do we rule out simply historical synteny conservation? So for the next few minutes, I will just talk about formal and informal criteria that allow us to make some tentative conclusions about the nature of these associations between various genes and CRISPR-Cas. So I'll begin with this example, which is one of the most prominent, most abundant. So it's a gene. So I indicate here the number of protein clusters. So when we identify we work with a great number of microbial genomes these days. So we always have to do clustering of genes to be able to analyze them and to understand what we are dealing with. And after clustering, we have 50 clusters, 50 families of this particular gene denoted core A, which is a membrane gene whose function is a metal-ion transfer of different ions. So this gene is found in association with a great number of and considerable variety of type 3 CRISPR-Cas systems. Moreover, it's not found alone. It's also found in a subset of these, in different subsets of these, associated with two other new CRISPR-associated genes which encode different kinds of nucleases. So we don't really know what is going on. But what these observations suggest to us is a functional membrane connection of type 3 CRISPR-Cas systems, where in all likelihood the core-A protein tethers the CRISPR-Cas system to the membrane and also engages these associated nuclease genes. It could be a phage DNA transporter. It could be a sensor of phage entry through membranes. It could be a regulator. It could be, finally, a cell suicide machine. This needs to be studied experimentally, but in a case like this, I think we don't have reasonable doubts about the functional relevance of the association of such a gene with CRISPR-Cas systems. Incidentally, serendipitously, in a previous work, we already, in a small group of bacteria, we already detected the horizontal mobility of this gene, whereby it's horizontally transferred together the 3B effector module and joins different adaptation modules and Cas6 genes, supporting the conclusion that these genes is really part of the functional modules of Cas genes. Proceeding along the list of these new predicted CRISPR-associated genes. Novel CARF domains. We already spoke a little bit about CARF domains. They are found in many CRISPR-Cas systems, primarily type 3. Once again, this is CRISPR-associated Rossmann fold, and as many people in the audience probably know, Rossmann fold is the most common type of nucleotide binding domain, and in the pioneering studies from Ginnick and Schicksny's lab, it has been shown that in type 3 it functions as an oligo-A binding domain. But in this analysis, we discovered a new variety of CARF domains which are very divergent from others and are linked to transmembrane helices in the respective proteins. Moreover, many of them are associated with distinct proteases of the lawn-like family. So probably we are observing here evidence of membrane-associated stress signaling linked to CRISPR-Cas systems. More membrane connection of type 3 CRISPR-Cas systems, in this case completely uncharacterized membrane proteins that are found, however, in many type 3 systems from actinobacteria, and again can be predicted to be involved in CRISPR-linked membrane signaling. So there are quite a few more examples like this. I don't have the opportunity to go through all of them. I just wanted to give you a visual impression on this slide of the complexity of the organization of type 3 CRISPR-Cas systems that exceeds anything that we see in other CRISPR-Cas variants. And here we have previously identified CRISPR-associated genes and quite many genes identified in the course of this project that I'm trying to describe now. So I think this is a big challenge for microbiologists that are maybe interested in CRISPR biology beyond adaptive immunity. Of course, this is not all. We shall continue along these lines, but now go away from type 3 and talk for a second about type 4. Type 4 is a special kind of CRISPR-Cas system, still very poorly characterized. They primarily, but although not exclusively, reside on plasmids, and they don't have any nuclease that could be responsible for interference. So they're found in quite many bacteria, and in plasmids from quite many bacteria, and as far as we can predict, they have nothing to do. They're incapable of interference, so they have to do something else. So what we observe through the CRISPRicity analysis is that many of these systems contain the protein of the cis-H family, which is a family of adenine nucleotide alpha hydrolases. We still do not know what these proteins do. Again, the best idea is some type of signaling function related specifically to type 4. More along these lines. Now from type 3 to type 4, from type 4 to type 1. What we observed in a particular subset of type 1e systems, again, found in many, primarily, actually even exclusively, in diverse actinomycetes. What we found here is the association of a particular variety of 1e systems with genes that encode the so-called stand NTPases. For me, this was a case of very welcome déjà vu because my colleagues and I did quite many years ago, did a comprehensive analysis and identified this family of very interesting ATPases and GTPases, which include, incidentally, the key signaling proteins of eukaryotic programmed cell death. There are many bacterial and archaeal homologs. No one really understands their functions. I hope that these findings that I'm describing to you now shall trigger a change in this situation. So we denote them stand signal transduction ATPases with numerous domains, because indeed, in addition to the ATPase domain, they typically contain a repetitive domain, such as TPR repeats, WD40 repeats, or others. And what we detect here is a stable association of these NTPases with a subset of 1E systems. Oh, sorry. So we do phylogenetic analysis, in this case of the Cas5 family from 1E systems, and we detect a number of distinct branches that includes Cas5 proteins from loci devoid of adaptation modules, which again suggest functions of CRISPR-Cas systems beyond adaptive immunity. So when we zoom in and perform the phylogenetic analysis of this particular branch, what we see is a very interesting picture. We see a particular branch that in which the 1E loci contain these stand NTPases associated with TPR domain, and another smaller branch where they contain stand NTPases associated with the WD40 domains. And we also see that in these branches, the Cas3 protein, which is responsible for interference, initially degrades, losing the helicase domain, and then disappears, apparently rendering these systems incapable of interference. So these are the observations on these NTPases associated with CRISPR, which really strongly suggest a link between CRISPR-Cas systems and either programmed cell death, so this could be defense without interference through programmed cell death, and or other signal transduction roles. Interestingly, some of these NTPases are also fused to caspase domain, which is a protease, which in all eukaryotes is a key effector of programmed cell death. So perhaps such processes remain to be discovered in bacteria. Very quickly, I will talk about a formal approach to differentiate functional associations from purely historical conservation or spurious co-occurrence. We try to do this through co-evolution analysis. So for each pair of loci, we build phylogenetic trees and derive distances from these trees. And we do this for effector module genes, for species tree, for 16 sRNA, and for the gene of interest. So if schematically, if we are talking about a real functional association, we will observe a strong correlation between these distances, like this. If we are talking about something spurious or purely historical synteny, we will not observe such an association. So as a control, there is this very strong correlation between Cas5 and Cas10 genes for type 3 CRISPR-Cas systems. And now we move to the core, to the newly predicted ancillary gene. And we see that the correlation between the phylogenetic history of core A and Cas10 is about as strong as that between Cas10 and Cas5, the bona fide Cas genes, whereas the correlation with 16 sRNA is quite weak, indicating the history of horizontal transfer in these of these genes. In other cases, we actually see signs of apparent vertical evolution for newly discovered accessory proteins, such as with the membrane curves. So to summarize this part, basically, we developed a systematic procedure for detection of CRISPR-associated genes with flexible parameters. We can play with this quite a bit and, of course, apply this to other functions in microbial genomes. We notice that the core Cas genes are already known. But that said, many diverged subfamilies, subgroups remain to be undetected and need to be discovered through the use of such methods and powerful sequence similarity detection techniques. We identified many new accessory genes and found that type 3 systems are, in a sense, most interesting and most complex. This is the home for the great majority of new genes with the emerging themes of membrane association and various forms of signaling. I don't know what is special about them. We have suspicions that maybe they may be actually ancestral to all CRISPR-Cas, and through that long history accumulated all these additional functions resulting in functional versatility. What I really want to emphasize is there is so much biology to be studied here. And here's another point that I want to emphasize, which is non-interfering CRISPR-Cas systems, those that contain no enzymes for target cleavage, and accordingly, by per force, do something else, something other than interference. This is the type 1E standard TPase containing glossary that I just described, program, so therefore single transduction is implied. These are the, so to speak, minimal type 1F systems carried by transposons that we described about a year ago, which may be involved in transposon integration. These are type 4 systems, about which I spoke a bit today, which may be involved in plasmid maintenance in ways that we don't still understand. These are inactivated type 5U systems, and I'm sure more of such systems are coming as we explore microbial genomes. I really want to bring home the point that it's oversimplification to think that CRISPR-Cas systems are just adaptive immunity systems in microbes. Apparently, they can do much more, and we have to figure out what. In the remaining few minutes, I will completely switch gears and speak a little about new roles for CRISPR-RNA. So far, we have been talking only about protein coding genes. A few moments about CRISPR-RNAs. So we know very well, it has become famous, that type 2 CRISPR-Cas systems use tracer RNA for pre-CR RNA processing and interference. And tracer RNA contain an anti-repeat region and form an imperfect hybrid with the CRISPR RNA. So we were interested to look more broadly in microbial genomes to see, to search for possible repeat-derived non-coding RNAs that may be somehow involved in CRISPR-Cas function. So we did a comprehensive search and came up with something that in part is a cautionary tale. Namely that something that looks like anti-repeat sequences that forms a decently stable hybrid with the repeat can be found in quite many places in the genome. So here we have the delta G distribution, and there are many of these quote-unquote anti-repeats in different places in the genome that are probably irrelevant. Here you have a highly stable repeat anti-repeat base pairing. Here we have also quite outstanding tracer RNA, but here you have something interesting. A little weaker than tracer RNA, but still quite stable anti-repeat upstream of the CRISPR array. And this is found in a variety of type 2A systems, some type 2B systems, and in type 6 systems as well. And in our work with Fang, it has been shown that in type 6A, this small anti-repeat RNA is highly expressed. We still have no idea what they might be doing, but the combination of high stability, low delta G of co-folding evolution and evolutionary conservation, and in this case, and expression shown in this case and a few other cases in Rodolphe Barango's lab, suggests to me that these things do have some function in CRISPR systems. And the final theme that I want to cover very quickly is search for CRISPR in bacteriophages. There are several examples of complete CRISPR-Cas systems found or identified in bacteriophages and apparently involved in anti-defense functions. And these provocative discoveries prompted us to do a comprehensive search. This analysis is still underway, but I will give you a quick and I think striking story. This is the story of conserved mini CRISPR arrays in bacteriophages. So these are several, this figure illustrates several phages of Streptococcus thermophilus, and all these phages in a particular region of the genome, in the region separating the genes for small and large terminase subunit, contains this unique feature, namely a CRISPR repeat identical to the type 2A repeat in the host, and then another either complete repeat or half repeat, and a unique spacer between them. There are no Cas genes and no tracer RNA in these phase genomes. But there are some interesting features here, suggesting that these arrays are likely to be expressed, namely good, strongly predicted promoters. Although the leader region and the Cas1 recognition motif are different from those in the host, suggesting that integration here might not be possible. Okay, then of course we asked the question, there are these interesting mini arrays in bacteriophages, what might be the spacers? And here we came up with a big surprise, namely that the spacers in these phage mini arrays target none other than the terminase genes in the same bacteriophages, except, not really, except they don't target the terminase genes in the same genome literally, but the terminase gene, because there are several mismatches that in all likelihood preclude targeting, but there is a perfect match to the terminase genes of closely related bacteriophages. And this is a repeated pattern that we observe in a variety of these bacteriophages. There are several other examples. So what we think happens here is phage wars. Apparently, the phage recruits the host CRISPR-Cas machinery to express these, the spacer of the CRISPR RNA from these mini arrays and preclude the super infection by the related bacteriophages. Remains to be tested experimentally. I think some experiments are going on in Sylvain Moineau's lab. So, the general conclusions. I think we fairly well understand the basic CRISPR mechanisms. And the most common CRISPR variants are known, although many new interesting ones for sure remain to be discovered. I'm not saying we have entered the phase of diminishing return in this basic CRISPR research, but just perchance we may not be that far from there. However, I think we are only starting to scratch the proverbial surface of the broader CRISPR biology, particularly connections with diverse microbial signal transduction systems. And also, there are many ecological, so to speak, role of CRISPR. There are many ways that CRISPR are used in the arms race by the host and by the viruses. All this remains to be explored, I think, for years to come. I have to thank people who participated in the work, the evolutionary genomics group at the NCBI, the main CRISPR guru, Kira Makarova, Sergei Shmakov, who discovered several new types and subtypes of CRISPR and designed the computational pipeline for CRISPRicity search, Yuri Wolf, who is not marked for some reason, but he is here, and who contributed a lot of thinking and a lot of computational wisdom to this research. Guillaume Faure, who explored the RNAs, And of course, the collaborators, they haven't showed much work done together with Fang. But without collaboration with Fang's lab, none of this would really make much sense. Konstantin Severino from Skolkovo and Rutgers, Joe Peters, with whom we worked on TN7 transposons, and of course, last but absolutely not least, the Arbor Biotechnologies company, in particular Winston Yan, David Scott, and Dave Chang, with whom we enjoy the vibrant ongoing collaboration. Thank you very much for your attention.
Regarding the phage wars, one can imagine that off-target activity could target the own phage genome. So would you expect that in this CRISPR-Cas, in this CRISPR system, the off-target activity is very low?
Biological reasoning clearly suggests that. And if you could notice, there are quite a few mismatches. If I recall correctly, actually, these are the same systems that have been studied in the classic 2007 paper by Barangu and Koverkas. And if you may recall the paper, which kind of started the whole thing, a single mismatch there eliminated the protective effect. These are those very same systems. So indeed, I suspect that there is practically no, and they're well protected. There are several mismatches. So I suspect there is practically no off-target.
Hi, what about the CRISPR array that was found like 3 years ago in a giant virus? Is it functional?
Okay, I should have a rule that you owe me $1 if you ask me that question. Because so many people do. There are no CRISPR arrays in giant viruses. There are no CRISPR arrays. There are only red herring. This so-called mimivir or mimivirea, whatever it is, it's kind of an interesting locus in mimiviruses. It contains repeats, which sort of trigger the thinking about the connection to CRISPR. And it may be involved potentially in defense. But if so, it remains to be studied. The experiments are inconclusive. If there is some defense function, it's certainly realized that the protein level, it has mechanistically or evolutionarily, has nothing, zero to do with CRISPR-Cas. So please don't talk about CRISPR arrays in giant viruses. There are no, for that matter, to bring home a general biological point, there are no CRISPR systems in eukaryotes or eukaryotic viruses. Over a glass of wine in a few minutes, I'm ready to talk why, but just let's remember this for now.
And another question, super fast. What about these ancillary proteins? Are there, most of them work at level of RNA or DNA? That defense or functionality of these neighbor proteins?
The accessory proteins that we have been talking. Most of them, or there are different ones. Most of them do neither. Most of them do something else, such as, say, membrane connections of their CRISPR systems. At least this is what we suspect, because they are definitely membrane proteins. Or signal transduction connections, such as we were standing in TPases. At least this is what we suspect, because they are definitely in TPases with signal transduction functions. Some of them are nucleases, and those nucleases, some of them are proteases, by the way. Some are nucleases, and those nucleases that we detect work primarily at the RNA level.
So speaking of wine, we have
Just one last question. Very quick. So we know the phages are expressing anti-CRISPR, to combat the CRISPR response from a bacteria. So now that phages have CRISPRs to attack other similar phages, do the phages have now anti-CRISPR systems as well?
You mean, is it the case that the same phage genome would contain anti-CRISPR proteins and anti-CRISPR, right?
Yeah, yeah.
The answer is I don't really know. Experimentally, there are no anti-CRISPR proteins have been identified in those phage genomes. It is difficult to develop a computational strategy to predict anti-CRISPR proteins, although we are actively working on that. And once we are done, I will be able to tell you with greater confidence. Right now, I think more likely these are exclusive, but I don't know for sure.
Okay, so Eugene will be around for the rest of the evening, so let's go out and have a drink, and you can continue to badger him with questions about CRISPR defense. Thank you, everyone.